Hello everyone, this is Dr. Deepa Dahar. I'm your botany faculty. Today we are going to discuss regarding this transcription process. Where does this transcription process happens? It happens inside this nucleus. This is our nucleus in which this double-stranded DNA is present. This double-stranded DNA is used for the formation of this single-stranded RNA. One process happens over here, another process happens for the formation of this long chain of proteins. These proteins are important part of our body. Since these are the building block of an organism and these proteins act as an enzymes also. You all are aware about the different types of reactions which happens and the requirement of enzymes for every process. Fine. So today in more detail we will be talking about this first process, that is transcription process, that is formation of this single stranded RNA. Okay. Okay students, so let's first have a look at some important points to be remembered. Okay. So we are going to discuss regarding certain processes like our DNA is double stranded in nature, right? That double stranded DNA is highly coiled in nature. Fine. That coiled structure has to be separated, can be used for the formation of RNA. The double stranded DNA will get separated into the single stranded DNA and any one of the strand will be used for the formation of RNA. The process of formation of RNA is called as what? Transcription process. Fine. That same single stranded RNA will be used for the formation of proteins. The process of formation of proteins will be called as what? Translation. These two important process has to be understood very clearly. DNA to RNA, RNA to protein, the whole process, these two process combined is called as central dogma of protein synthesis. That means protein synthesis could not happen without transcription process. First transcription has to be get completed, then translation has to be followed. Okay. One more important process, that is called as replication. When the body needs to form extra copies of DNA, the same DNA, that double stranded DNA will be used for the formation of its replica, or you can say its copy. So the process of formation of another copy of DNA is called as replication. Fine. Now, what is transcription? Nothing but what? Formation of RNA from DNA. But what does this word mean? Transcript. Script means what? Certain information. You all are aware about that our DNA, this double stranded DNA carries certain genetic information. That genetic information has to be translated. So, transfer of certain genetic information, right? Transfer of certain genetic information is called as transcription. Script means information. Transcript means transfer of that script in the form of RNA. This RNA will be used for the formation of proteins. The process is called as what? Translation. Translate means what? Translation of certain genetic information in the form of proteins. So, for these processes, certain enzymes are required which will do transcription and translation process. Is it clear to all of you that central dogma of protein synthesis states that formation of proteins with the help of RNA and DNA. Fine. Now, coming to the transcription process in detail. What are the requirements for the transcription process? So, let's talk about this transcription process now. We need a cell. From that cell, we need nucleus. Why we need nucleus? Because in this nucleus, our DNA is situated. Double stranded DNA, which is in the coiled form, that is situated inside the nucleus. And this transcription process happens inside this nucleus only. These are the basic things, but the important things which we need for transcription are, for the starting of process, we need initiation factor, nothing but called as sigma factor. Since the process has started, the process has to be stopped. So for termination of process, we need a termination process. To stopping the process, we need termination factor, nothing but rho factor. Important point to be noted that we need sigma factor to start the process. We need rho factor to stop the process. But along with this sigma factor for starting the transcription process, we need this enzyme. What is that enzyme? That is RNA polymerase enzyme. Why RNA polymerase enzyme? Because the process of formation of RNA is happening. So the polymerase enzyme has to be RNA. Suppose if the process is formation of DNA from DNA only, that means we need another copy of DNA. So DNA is used for the formation of its replica. So the enzyme will be used called as DNA polymerase enzyme. Why? Because DNA is being formed. Now in this process, since RNA is being formed, so the enzyme will be RNA polymerase enzyme. Correct? But since this process is happening in all organisms, but little bit difficulties come in little bit complex organisms. Like in case of prokaryotic organism, we need only one type of RNA polymerase enzyme, because the organisms are not that much complex, and DNA in those organisms is not enclosed within the nucleus. It is freely suspended in the cell. So the process is little bit simpler in case of prokaryotic organism. Correct? Now, in case of eukaryotic organism, since the organisms are complex and the DNA is situated inside the nucleus, so for this we need three different types of RNA polymerase enzyme. RNA polymerase enzyme 1, enzyme 2 and enzyme 3. Okay? Three different types of RNA polymerase enzymes are being used. The process will be same, but the outcome will be different. What is the outcome for three different types of enzyme? See, RNA polymerase enzyme 1 will be used for the formation of rRNA. What is rRNA? rRNA is ribosomal RNA. You all are aware about these three different kinds of RNA. Fine. What is mRNA and what type of enzyme is being used? The enzyme 2. RNA polymerase enzyme 2 is used for the formation of mRNA. What is mRNA? mRNA is messenger RNA. Our mRNA is what? Messenger RNA, which carries message from the nucleus. Okay? Now, RNA polymerase enzyme 3 is used for the formation of tRNA. What does this mean? tRNA is transfer RNA. So three different types of rRNAs, are formed with the help of three different types of polymerase enzyme. Correct? But in case of eukaryotic organism. In case of prokaryotic organism, only one type of RNA polymerase enzyme is used. Okay, so we need nucleus, we need DNA, we need initiation factor, we need termination factor, we need RNA polymerase enzyme. Fine. Coming to the process of transcription. This transcription process involves three very easy steps. See, the process has to be started. So the process is called as what? Initiation. We need a cell. From this cell, DNA is used for the process. So the process which starts the reaction, when the process starts, so the step will be called as initiation. Correct? So for the initiation process, what you need? Just now we have discussed. Right? What? That is sigma factor, correct. And second one is RNA polymerase enzyme. Okay. So what exactly happens in this process? RNA polymerase enzyme will recognize certain sequences on the DNA. Okay. Since the DNA is highly coiled in nature, it has to undergo unwinding process, because only one strand of DNA, any one strand of the DNA will be used for the process. So RNA polymerase enzyme, this RNA polymerase enzyme will choose one position or one area of the DNA. What kind of area will be used? See, That area is called as what? Initiation site. That initiation site can be of TATA rich regions. These TATA regions are called as Tata box region. These are the initiation sites for the transcription process. So, where this Tata box is present, RNA polymerase enzyme will recognize that Tata box, and after that it will start the process. See, this point is to be remembered with related to NEET point of view. For the board point of view, only you need to remember the initiation site. Okay. So, this RNA polymerase enzyme will recognize the initiation site and will move in the forward direction and keep on unwinding the DNA. This RNA polymerase enzyme will work with the help of sigma factor. Okay. Now, coming to the second step. What is our second process? Second process is called as what? Elongation. This second process is called as what? Elongation. What happens in this process? This RNA polymerase enzyme will move in the forward direction. RNA polymerase enzyme will move in the forward direction. Simultaneously, it will keep on attracting these nucleotides. You all are aware about different types of nucleotides present in a cell. What type of nucleotides are present? That is A, T, G, C. A stands for adenine, T for thymine, G for guanine, and C for cytosine. Correct? And again, you all are aware about that A will always pair with T by double bond, G will always pair with C by triple hydrogen bond. This is in case of DNA. But what happens in case of RNA? In case of RNA, this T is replaced by U. That means in case of RNA, A will pair with U with the help of double hydrogen bond. This will happen in case of RNA, and this will happen in case of DNA. Correct? Rest G triple bond C will be same in case of RNA. So now what this process is doing now? This RNA polymerase enzyme is attracting these complementary nucleotide. Complementary nucleotide means if A is present, RNA polymerase enzyme will attract U in case of RNA, because the process is formation of RNA. And this G will attack whom? C. Fine. As the process moves forward in the forward direction, this RNA polymerase will keep on attracting different complementary nucleotides. But these nucleotides cannot come alone. They need certain amount of energy. Okay, so that energy will be present in the form of 3-phosphate group. So every nucleotide is attached with 3-phosphate groups. That is ATP, UTP, CTP, GTP. That is adenosine triphosphate, uracil triphosphate, and so on. Fine. Now these triphosphate groups are attached with every nucleotide so that they need some amount of energy. They can move from matrix to the DNA. Fine. These all nucleotides, these all 4 nucleotides can be called as NTPs. Nucleotide triphosphate. 4 different types of nucleotides are involved. Okay. In short, it can be called as NTPs. Now, the elongation process. Elongate means what? Moving forward. Elongate, extension of the process. So this RNA polymerase enzyme will move forward and help in the formation of RNA. This RNA, see, this RNA is being formed over here with the help of complementary nucleotides attached on the DNA. Simultaneously, the older formed RNA is being released into the matrix, and the previously unwinded DNA will undergo coiling process. Fine. The forwarded DNA will undergo unwinding process and the previous DNA will undergo coiling process. Fine. This is the elongation process. Elongation happens till the termination region comes, till the stop point comes. Okay. Coming to the third process. What is third process? Of course, termination. We initiated the process, we elongated the process, now we need to terminate the process. We need to stop the process. So, this third process is called as what? Termination process. For termination process, what you need? Which factor you need? You need rho factor. So, rho factor will come and stop the process at certain point. And at this point, RNA polymerase enzyme will be released. It will be free from the DNA and the rho factor will be released. And finally, our RNA will be released in the matrix. Now, one point to be noted that this RNA is having direction from 5' to 3' direction. Remember, every RNA has to be formed in 5' to 3' direction only. You all are aware about these directions, correct? Now, that means the DNA which was used for the transcription process has to be from 3' to 5' direction. Correct? Any one of the strand can be used which is in 3' to 5' direction. Of course, it will be complementary. So, if one strand is from 3' to 5', so another will be from 5' to 3' only. Correct? Just a second. Yeah. So, we got this RNA. Fine, this RNA is having certain nucleotides, certain information which is being copied from DNA. Now, this RNA will be used for the formation of proteins in case of prokaryotes. Why did I said that? Because in case of eukaryotes, the process has not stopped yet. It has to undergo further processes. Complex organisms, complex process. But in case of prokaryotes, the process stops over here only. Fine. Now, since we got this RNA, but in case of eukaryotes, this process takes place further. Now, in the previous slide, as I have shown you three different diagrams for the three stages. That is for the understanding purpose, but at the exam level, for exam, no need to draw three different diagrams. You need to draw this one diagram only. In this one diagram, you will show double-stranded DNA, and this double-stranded DNA will be having RNA polymerase enzyme attached at one certain position. So this RNA polymerase enzyme will move in the forward direction. Okay. And one template will be used for the formation of this RNA. Fine. Now, since it is looking like an open position, it is looking like a bubble. So this structure can be called as what? Transcription bubble. So, where the transcription is being happening. You need to show this unwinding of DNA and this is a rewinding of DNA. And this mRNA has been released in the matrix from the DNA. It got detached from the DNA. It will keep on detaching and it will get, finally at the end it will get released in the matrix only. Fine. Now, coming to the further processing of our RNA. The RNA which we got is not mature RNA. That RNA is called as heterogeneous nuclear RNA. That is immature in case of eukaryotes. So, in short, it can be called as what? hnRNA. So, this hnRNA has to undergo different processes. But in case of eukaryotes, in case of prokaryotes, no further processing is required. In case of eukaryotes, it has to undergo three different processes. Again, the three different processes are very easier in nature. What are those processes? Let's have a look at these processes. Now, first step, the first process which happens is capping. What does this capping means? Where we use a cap, if you are supposed to wear a cap, you will wear it on your head. That is at the start point of your body. So, I need to place a cap on the RNA. So, I need to choose a starting position of the RNA. What is that starting position? That is the 5 prime end. The cap which has to be added is to be methyl guanosine triphosphate. Remember, very important thing to be remember, that is methyl guanosine triphosphate cap is added at the 5 prime end. In exam, in multiple choice question, it may be asked that cap has to be added at which position? It has to be added at 5 prime position. Or what kind of cap is to be added? See, you have to write it like this way only. Methyl guanosine triphosphate. Okay, this is the cap which we have added at the 5 prime end. This is 5 prime end methyl guanosine triphosphate cap. So we are done with the first process, that is capping. Coming to the second process called as splicing. As you all can see, I have shown here different regions showing two you can say spots alternating with each other. One is exon, another one is intron. These are placed alternatively with each other. What does this you can say name means? These are nothing but having certain sequences, correct? Exons will be coding for the proteins, but introns does not code for proteins. Introns are the interrupting sequences. Interrupting whom? Interrupting exons, because they are not coding for proteins. See, after transcription, translation process has to be followed. That is for formation of proteins. For that, I need continuous sequence of coding nucleotides, which are coding for the proteins. So for that process, I don't want these introns because they will interrupt the process of translation. So they have to be removed because they are of no use. They are not coding for proteins. For the timing, I need to remove these introns. You can see this RNA structure starts with exons and ends with exons, and in between there is presence of introns. So these introns has to be removed and every exon has to be joined together again. Okay, so the process will be called as what? Splicing. This splicing process happens with the help of enzyme spliceosome. Okay, spliceosome. The process is happening with the help of enzyme spliceosome. Splicing with the help of spliceosome enzyme. Fine, we have removed introns also. Now you can see these certain ball-like structure, ring-like structures appears on mRNA structure. Sometimes we may observe inside nucleus, when we observe this in a microscope, these ring-like structure may appear, but at the mature stage we cannot see these. Of course, you cannot use compound microscope, right? For observing this mRNA, we need another type of microscope. So after removal of this intron, I got this continuous length of exons, that means continuous sequences which are coding for proteins. Coming to the third process called as tailing. What does this tailing means? Tailing means what? Where is tail present? At the end. So we need to add tail at the last position of RNA, that is at the 3 prime end. So here at the 3 prime end, long tail is being added. This tail is of long adenylated residues, continuous AAA sequences. Hence the tail is called as what? Poly A tail. Poly means what? Many. A means what? Adenine. So many adenylated residues will be added at the 3 prime end. That is over here. Fine. These nucleotides, these adenylated residues can range, like they can be from 50 to 250 in nature. They can be small, they can be little bit large also. So a long tail, that is poly A tail, has been added at the 3 prime end. We are done with 3 processes, that is capping, splicing and tailing. Remember, sequence has to be in this form only, because first capping process is being done, second splicing has to be done, at the end tailing has to be done. Fine. Why these processes are happening? We can easily understand splicing process because we don't need introns, because they are interrupting sequences, they are not coding for any proteins. But why this capping and tailing is being done? Because after this transcription process, our DNA has to take an exit from the nucleus. While taking an exit, this RNA may get attacked by different types of enzyme. So to provide the protection and to provide more stability to our this RNA, we need to add cap and tail. Cap at the 5' end, tail at the 3' end. Fine. So why this process is being done in case of eukaryotes? Because when RNA takes an exit from the nucleus, what type of RNA we are discussing is the type of mRNA. This is what our mRNA which is being formed, that is messenger RNA. Why messenger RNA? It is carrying us certain genetic information from the DNA, because we have used DNA, one template of the DNA, and that is having certain information which is being transcribed in the form of RNA. Correct? So this is mRNA which take an exit from the nucleus. While taking an exit, it may get attacked by certain types of enzyme, it may get degraded. So for the protection purpose, for providing more stability, cap and tail has been added. I hope this much is clear to all of you. Coming to this structure of mature RNA, that is mRNA, that is messenger RNA. Now you can observe this structure. See, this is protein coding region. That means it contains only exons, correct, which will help in the formation of proteins. Now at the 5' end, methyl guanosine triphosphate cap is being added. Correct? At the 3' end, poly A tail is being added. But you can see certain sequences present after cap and before tail. What are these sequences? These are called as untranslated regions. These will be always present at the start and end of protein coding sequences. Fine. But these are called as untranslated regions. That means they will not take part in the formation of proteins. Fine. Then why these sequences come? At the start of the process, I told you that RNA polymerase enzyme will recognize certain sequences to start the process, like Tata box regions. So those regions got transcribed but will not take part in the protein synthesis. So since they are not carrying protein synthesis process, so they are called as what? Untranslated regions. So this is our mature mRNA structure which we got after transcription process, and this will go out of the nucleus now. Fine. See, this is our nucleus where transcription process has been done. This transcription process helped in the formation of mRNA. From the nucleus, with the help of these pores, this mRNA will take an exit and move to the cytoplasm. Correct? So see, that's what I said, that when it takes the exit from the nucleus, it might get attacked with the different types of enzymes. So protect our mRNA, the process of capping and tailing has been done. Fine. So this mRNA will be used for the next process called as translation. What does that translation process means? That is formation of proteins from this mature mRNA. Fine. So mature mRNA take an exit from the nucleus and moves into the cytoplasm for the next process called as translation. That is formation of protein. That is the main result. This translation process we will be discussing in our next class. I hope this much is clear to all of you. Thank you so much for listening me. Okay. Bye-bye. See you. Have a good day, and we'll meet you in the next class explaining more concepts with more colorful diagrams. Bye-bye.